Hello. Hello. Samuel sir, I'm audible. Hello.
Yes, sir. Yes, sir.
Okay, good morning. A very good morning to everyone to present here with warm greetings. And it gives me a great pleasure to extend warm welcome on behalf of Sree Sastha Pharmacy College conducting one-day webinar on role of pharmacy education in drug discovery and development in the present and future. Know your brand as a pharmacist. This is our sixth webinar series. Myself, Dr. Dhanapal, principal of Sree Sastha Pharmacy College located at Chennai. Our institution running by A.M. Kanniyappa Mudaliyar and A.M. Jambalingam Mudaliyar Educational Trust. Our trust is managed by Professor J. Karthekeyan, B.E. M.B.A. He has deeply committed to the responsibility of training of students and staffs in terms of attitude, knowledge, and skill set to face the real practical life and professional life. Our trust is also running various institutions like engineering, nursing, arts and science, polytechnic, and educational colleges. I would like to sincere thanks to our chairman to permit us to conduct the webinar series from our college. Our college offering diploma as well as B.Pharm courses. I feel privileged to extend my warm welcome to the entire participants, including principals, professors, associate professors, assistant professors, and industrial persons and pharmacists, my dear students from various across India and around the world. Success is not a key to happiness, but happiness is key to success. I would like to introduce today's our presenter, Dr. Mahendran Sekar, Associate Professor of Faculty of Pharmacy and Health Sciences, University of Kuala Lumpur, Malaysia. He has completed his undergraduate and postgraduate and PhD at Tamilnadu Dr. M.B.A. Medical University, Chennai. He received many awards and research funds for his career. My sincere thanks to the presenter for his acceptance of my invitation for this webinar. Last and not least, I welcome to the Lord and His presence for this webinar. Enjoy next 1 hour for this webinar to learn something from that is research and development of drugs. Thank you, one and all. Please listen. I hand over to Dr. Mahendran Sekar.
Yeah, good morning to everyone, and thank you very much for Dr. Dhanapal sir for the kind introduction. So I take this opportunity to thank Sastha College of Pharmacy, Chennai, for inviting me for this webinar series 6.0. So can you please allow me to share my slide?
Okay, sir.
Yeah. Is it visible?
Yes, sir. Yes, sir.
Okay, thank you very much. Okay. So the topic which I was chosen for today's presentation is role of pharmacy education in drug discovery and development in the present and future. Know your brand as a pharmacist. So why particularly I chosen this topic because as I know in this webinar most of them are undergraduate and postgraduate students, and some of them are researchers. Maybe some of them might be working in an industry. But this presentation I mainly focused on the future pharmacist who is studying undergraduate and also the postgraduate studies in pharmaceutical sciences. So as you know, in pharmaceutical sciences or pharmacy program, we study lot of subject. Because pharmacy is a multidisciplinary course program which including chemistry, pharmacology, study microbiology, biotechnology, phytochemistry. So it is a multidisciplinary subjects are included in the pharmacy program. But when you study a pharmacy, so you must know how each and every subject which are present in the curriculum, how it is related to the drug discovery development and further process, so especially the clinical trials. So that is what my presentation I am going to explain. Hopefully this presentation is it clear?
Mahendran sir, not clear, sir.
Now
Echo
is it okay now?
Echo is there.
Echo.
Two mic is on.
No, only one mic on. Just I'm using my. Okay.
Okay.
Is it okay? Clear now?
Okay, sir.
Okay, thank you. So as you know, the use of drugs in daily life has been increased in recent years, either due to the emergence of new diseases, as you know, in the recent 10 years, if you look at there are more number of new diseases, which including started from Ebola, SARS, Nipah virus, then Zika virus, then recently we are facing COVID-19. So there are so many new diseases and also chronic disorders, including diabetes, cancer, and as well as cardiovascular disorders. So the drug cannot be avoidable for everyone. It is supposed to be taken any time in your lifetime. You might be taken. So on the other hand, the presence of lifestyle interventions, which including harmful pollutants and microorganisms. As I mentioned, microorganisms which including bacteria, fungi, and virus. So because of that, there are so many new diseases. Now even COVID-19 because of virus. So as a pharmacist, we need to find the solution for those diseases. We need to find the treatment for those diseases. So especially novel and safer pharmacological treatments are heavily needed for treatment of all these diseases, including the chronic disorders and diabetes, cancer, as well as cardiovascular disorders. So as a pharmacist, we cannot run away actually from development and discovery process. Because since we are the pillars actually in various development centers, including public, private and foundations. And as you know, the definition for pharmacy. Actually, there are so many definitions are available for pharmacy. But here I took one of the definitions, which is mentioned as pharmacy is an art or practice of preparing, preserving, and compounding medicines and dispensing them according to prescriptions. So if you look at this one, there is no word about mentioning about discovery and development or maybe in clinical studies. So just started with preparing the medicine and preserving, compounding, then finally we are dispensing and counseling given to the patients. So this is what mentioned in the most of the definitions. So considering the current changes in our syllabus, and our pharmacy is a multidisciplinary subject we are studying during the pharmacy. So the definition must to be changed now. So especially we need to include something to the drug development and discovery. So pharmaceutical researchers have been involved throughout the drug discovery process. As you know, it started from the identification of a new molecule until the final drug. When it comes to the market, the pharmacist play a major role actually. So that is why I mentioned that drug candidate from preclinical research to clinical stage, bench side to bedside. So it is started from the lab work done by the pharmacist. Then finally it goes to the customer, that is for patients, from the retail pharmacy. So from the beginning until the end, pharmacists play a major role in the drug discovery and development process. So as you know, the courses that are offered in most of the countries, including India and even in other Western countries, these are all the programs which including Diploma in Pharmacy, Bachelor of Pharmacy, we have PharmD program, Master of Pharmacy and PhD in Pharmacy. After completion of pharmacy course, these are all the career choice that our students normally they get it. Either they work as a retail pharmacist in community pharmacy, or they work as a hospital pharmacist or clinical pharmacist in the hospital, or either they can go for academic as an academician or lecturer. Then they can go for industry and work as an industrial pharmacist. So there are so many divisions are there in the industry. Either you can work in a production or quality control or quality assurance, regulatory affairs. But here in my focus, I am mainly focusing on research and development. So what if you are really interested in research and development, which is R&D, then you must be linked with what are the subject that you study in our B.Pharm course. So that is what my presentation is mainly going to focus on. So as you know, drug discovery process every day, if you look at in the market, there are 100,000 of molecules are tested in every day for the drug discovery process. So cost of this screening is very, very expensive, because starting from the identification of compound and doing all the laboratory work, then it is need to be tested with animals, then followed by clinical trials. So it is a very expensive and time consuming process. So what, as a pharmacist, what we need to do actually, we need to minimize this process. How we can minimize this process? By identifying or developing a new technologies or techniques, so that we can optimize all these steps and which are involved in the development of new drugs. Otherwise, if you don't optimize this kind of process, definitely this process cannot be minimized, because any drug molecules, if it comes to the market, means it takes minimum of 15 years of time, 10 to 15 years of time. So as you know, there's a subject which you study in B Pharmacy, including pharmacognosy, pharmaceutical biotechnology, pharmaceutical chemistry, pharmaceutical analysis, pharmacology, and pharmaceutics. So these are all the main subject which comes under the preclinical stage of drug discovery and development process. So here I mentioned pharmacognosy, means it is not only about pharmacognosy. Pharmacognosy, which including phytochemistry, natural product chemistry, this all comes under pharmacognosy discipline. Then similarly, pharmaceutical biotechnology means you study about biotechnology, you study about microbiology, immunology, and chemistry starts from organic chemistry, advanced organic chemistry, inorganic chemistry, physical chemistry, then followed by medicinal chemistry, then analysis, normal pharmaceutical analysis 1 and 2, or maybe modern pharmaceutical analysis, where you study about all the instrumental analysis, then pharmacology, then pharmaceutics. Also you have various subjects like physical pharmacy, pharmaceutics, then followed by industrial pharmacy, and also you may study about the subject which comes under pharmaceutics department, like bioengineering and biopharmaceutics, like that. So so when you study those subjects, what I am telling is you need to link with how these individual subjects are related to the drug discovery and development process. So this is all about the preclinical stages. So once it moves to the clinical stages, the PharmD students play a major role, and also pharmacovigilance, regulatory affairs, pharmaceutical management. So this all comes under clinical stage. So I'm not going to cover more about the clinical side. So I will cover more about the preclinical side, every individual discipline, how it is related to the drug discovery and development process. And I'm also going to explain about what are the recent techniques or technologies, or maybe the methods which you can follow, which you can adopt in your research, so that you can minimize the drug discovery and development process, and we can see the product in a very short period of time. See, for example, in COVID-19 also, we come up with the vaccine within 1 to 2 years of time. So because it is a global issue, so everyone started up doing that research, and they come up with so many new innovation techniques or technologies they have been used, so that they can able to come up with the vaccine in 1 to 2 years of time. This is called as minimizing the drug discovery process. So why cannot we can minimize the process for the other drugs as well? Means treatment for other diseases like chronic disorders, like diabetes, cardiovascular disorders, and cancer. So this is what we need to think as a pharmacist. So this is the overall global view of role of pharmacy in drug discovery and development. As I mentioned, it always starts from the finding a new target against a disease or disorders, which is the foundation of drug discovery and development process. So here I mentioned that there are some omics. So several omics, which including genomics, metabolomics, lipidomics, proteomics. So these are all the methods and screening procedure used in the biomarker prediction, because we need to predict the biomarker before we move into the preclinical studies. So that is what we need to conduct basic research on physiology and pathology, then biomarker prediction and biomarker identification by using all this omics technology, which including genomics, metabolomics, etc. So after that, upon validation of the biomarker, what we do normally as researchers, we move into the chemical libraries which are evaluated against a target by using high throughput screening, which is the preclinical stage. So what we do in the preclinical stage, we normally conduct in vitro studies and in vivo studies. So in vitro studies, which including chemical methods, or maybe you may use some kind of cell lines to predict the biological activities. Similarly, once you find your molecule or the lead molecule which is having some potential biological activities in the in vitro studies, then those molecules will be taken to in vivo studies. So in vivo studies, which including the animal models, where we study about pharmacological activity and also the toxicity studies. So, so after this, which moves into the ADME properties, which including absorption, distribution, metabolism, and excretion studies, about the PKPD, like pharmacokinetics and pharmacodynamic study also will be conducted in the preclinical stage also. And whenever you choose the animal models for preclinical studies, we must be taken care of something, because finally, whatever the molecule that we are testing against animals, finally that is to be administered to the human, right? So whenever you choose the model for conducting animal studies, you need to choose the model which mimic the human being, which is because later on we are going to administer to the humans. Then after this, the formulation, optimization, and investigational new drug prerequisite requirements will be done. So once that has been done, then after that it moves into the clinical stage, which is phase 1, phase 2, phase 3, as you know. So phase 1 clinical trial normally conducted with the healthy volunteers to just check about the effectiveness, not effectiveness. It is just to check about the safety of the particular compound or maybe the molecule. So once you find that particular molecule is safe to the humans, then it moves to the phase 2 clinical trials, which is tested with a small number of patients. So where we can identify or determine the effectiveness of the particular molecule. Then once this has been done, this comes under PKPD, like pharmacokinetics and pharmacodynamics. So what the drug does to the body and what the drug does to the body. So this here only the PharmD students, they play a major role, especially. So when we conduct ADR monitoring, and also we associated with the clinicians to check about any toxicity producing whether that particular molecule or not. Then it moves to the phase 3 clinical trials, where we do the study by using large number of patients. So once that has been done, then that only it moves to the new drug application. So that moves into the FDA review. So here in the new drug application submission, so the regulatory affairs play a major role. So everywhere, whatever the subject you study, everything I have been included here. So if you look at this one, you can determine and you can say clearly how each and every subject, every discipline, or maybe the specialization, how it is related to the drug discovery and development process. Then finally, it moves into the after the FDA review, it moves into the final clinical trial stage, that which is phase 4. So phase 4 is something like a post-marketing surveillance, PMS. So here only once this one pass through, then only that is approved by the FDA. Then you can see in the before the phase 3, the drug will be available in the market, in the retail shop. Then the post-marketing survey will be conducted. That is considered as phase 4 clinical trials. So here, if you look at the other diagram which I mentioned in the left-hand side, so you can see the pharmacognosy, where you can get all this molecule either from the natural product or maybe from the microorganism. Then how you can test those molecules, whether it is how you can identify what is the type of molecule or the characterization, or qualitative and quantitative analysis can be done, pharmaceutical analysis. Then pharmaceutical chemistry, their main role is to determine the chemical structure and synthetic procedure. They need to come up with the synthetic procedure, how we can synthesize, how we can modify the chemical structure to improve the pharmacological activity and to reduce the side effect. Because our main aim is we need to give a high efficacy and low toxicity drugs into the market. So that is the main aim. Okay. So here in my presentation, I'm going to focus on 6 major disciplines which we study, which normally the students study in the B.Pharm program. I will start from pharmacognosy. So pharmacognosy, as you know, it is about the natural products. So as you know, natural products play a major role in the drug discovery and development process since ancient times. And as you know, why many researchers are interested in natural product is because natural product, as you know, this is a natural product produce low toxic and high efficacy when compared to the modern drugs. And we must know how the process will be to the drug discovery and development, starting from the collection of the plant material. So once you have been collected the plant material, immediately we need to get authentication from the botanist, because that is very important, because which plant that you obtained from, that is very important. So you need to give a correct information, means you need to get authentication from the botanist. So once you get the authentication from the botanist, then we need to keep drying of the plant material. So once the plant material is completely dried, then we grind the plant material into a coarse powder form and we select the solvent. So when you select the solvent system for the extraction, so you need to be select appropriate solvent, especially as you know, most of the secondary metabolites which are polar soluble in nature. So when you choose the solvent for the extraction, you need to choose the solvent which is highly polar in nature. So if you look at the polar in nature solvents, means either you can use water, methanol, ethanol, those kind of solvents can be used to prepare the extract. Then once the extraction has been done, means you can use, there are so many methods to conduct the extraction, like Soxhlet extraction method and maceration method, and there are so many new methods also available, which including microwave assisted extraction, supercritical fluid extraction, sonication method. There are so many new techniques are available which you can adopt to extract the plant material. Then once the extraction is done, then it moves into the concentration of the extract. So how you concentrate the extract? Normally by using the rotary evaporator under controlled temperature and pressure. So once the concentration has been done, then it moves into the next stage, that is isolation and purification. So isolation and purification normally done by using the chromatographic techniques. So as you know, isolation can be done by using column chromatography, and recently we have the flash column chromatography, where the manual work is reduced and the technology is used. So what if you use the flash column chromatography, you can isolate the compound in a fast manner, and at the same time you can also compare the fraction, the compounds which is present in the fractions, which compare with the some of the natural products library, means the chemical information which is available in the system, that can detect and tell what type of compounds is present in your each fractions. So this way we can minimize the process of isolation and purification. So once you are done isolated and purified and you identify it is a single, because the disadvantage of the natural product is when you do the isolation, you can get very minimum quantity. Either you get very few milligrams, like 10 milligram or 20 milligram of compound. With the 20 milligram of compound, definitely you cannot do any preclinical studies. So what normally the present researchers are doing, once the isolation has been done, once you identify the chemical structure, then you just pass it to the medicinal chemist who can synthesize that particular compound. So once they synthesize in a large quantity, then you can move into the in vitro and in vivo studies. So that way it moves into the pharmacological studies finally. Okay, so this is the normal process of natural product researchers, and these are all the things that you study normally in pharmacognosy, phytochemistry, and natural product chemistry. So as you know, pharmacognosy is now prepared to meet the current and future demands of drug discovery and development process. And we currently have so many innovative methods and technologies, especially the recent techniques and the instruments which are currently available in the market, that are very sophisticated, and you can get accurate results. So that is why my suggestion to the current researcher or maybe the students, you need to adapt with the latest technology so that you can minimize the time, okay, in the drug discovery and development process. And also which is expected to emerge as a result advances in drug target elucidation and lead structure discovery. So even the structural elucidation also, now we have the many softwares where you can do comparison of your spectrum as well. So that is why you need to adapt with the new technologies so that you can minimize the time in each and every step. See, for example, starts from the extraction, we have so many new methods. Similarly, isolation, you need to follow the flash column chromatography method to reduce the time. Once that has been done, then in vitro studies, before moving into the in vitro studies, you have in silico methods also. So you can adapt with in silico studies to select the particular compound to go into the in vivo or in vitro studies. So it is not necessary that you need to take all the molecules to move into the in vivo studies, because in vivo studies is very expensive and we supposed to use the experimental animals. So to avoid that one, we need to select the compound. How we can select the compound? Now we have the in silico studies where you can identify the pharmacological activities based on the binding efficiency with the enzymes or maybe the drug receptors, so that you can predict earlier to move into the in vitro or in vivo studies by using the computational or bioinformatics methods. So new technologies, as I mentioned, which including the combinatorial chemistry and also the computational chemistry, means which I mentioned all the computerized things, which including bioinformatics, in silico studies, or maybe the molecular docking studies. So in that way, you can minimize the time-consuming process, especially in the isolation, then following by the pharmacological study. So before moving into the pharmacological study itself, we can predict what may be the predicted biological properties of that particular molecule or maybe the compound. So these technologies, which can compensate the limitations against natural products and provide an exceptional opportunity to reclaim their place as a major source of drug discovery. So as I mentioned, the currently available drugs in the market, I think mostly around 30 to 40% of the drugs are mainly obtained from the natural source or derived from the natural source. So that is why still natural source is one of the main source where we can get the drug molecule to treat many diseases, even including COVID-19 also. Now there are so many researchers, they have been come up with a new molecule, especially from the natural products. So this all about the pharmacognosy. So when you study a pharmacognosy subject or maybe phytochemistry or maybe natural product chemistry, you must know how we can minimize the process to develop a new drug into the market. Similarly, what are the new techniques that you can use it? That is what I have been mentioned. Then next we will move into the pharmaceutical technology. So here in pharmaceutical biotechnology. So here pharmaceutical biotechnology, as I mentioned the previous one, pharmacognosy means the natural products obtained from the natural source, means especially from the plant material. So here the microorganism also is considered as one of the natural product, which is naturally obtained from the microorganism. So this also is considered as a natural products. And here the process normally starts from the extraction and isolation of the microbial strain, especially the whole cells or cell components or modified cells, which can be extracted from the microorganism. So that is considered as a primary, secondary, or other metabolites and also the components. So as I mentioned, the compounds or the drug molecule, it is not only obtained from the plant material, also we can obtain from the microorganisms. So one simple example, if I would like to give for this one, is Penicillium notatum is a fungus which has been initially used to isolate the penicillin, which is the first antibiotic introduced into the market. So penicillin was obtained from Penicillium notatum, not from the plant material, not directly synthesized. So similarly, there are plenty of compounds or plenty of drug molecules are available from even from the microorganisms, either from bacteria or maybe from other pathogens, something like fungus. We can get so many new molecules. So these molecules can be even the microorganisms can be used as a food additives, food preservatives, or it can be used as a starting material for the synthesis of some of the new drug molecule. So where the pharmaceutical chemistry discipline play a major role. So once you identify the chemical structure which is present into the microorganism, that molecule has been taken care and taken into a starting material to synthesize some new molecule which can be used for the treatment of disorders. And also directly there are certain primary or secondary metabolites which can be converted into a vaccine, drugs or probiotics. Okay. So similarly now even for COVID-19 also there are many research is going on. The metabolites which are obtained from the microorganisms which is directly converted into a vaccine form. So apart from that, the last one, we can also develop therapeutically useful proteins from the microorganisms. So these are all the things that normally we need to think about in the drug discovery process, where the biotechnology subject, which including microbiology, immunology, biotechnology, whatever you study in Bachelor of Pharmacy, so you can directly link with these kind of things, so that when you study that particular subject, you can easily understand why you are studying that particular subject and how that particular subject is useful in the drug discovery and development process. Okay. So now researchers even they are involved and working on some of the converting DNA sequence data and information that also can be used to change the drug discovery and personalized medicine. Now there are many researchers, they are focusing on the personalized medicines where the DNA and RNA play a major role. So here actually I mentioned about the omics like genomics, where the genome sequencing, and epigenomics where the DNA methylation is involved, and transcriptomics where the RNA sequencing is involved. So based on this, we can develop and we can come up with the personalized medicines. You can discover a new personalized medicine. So where this and all you study normally in the biotechnology subject about genomics, epigenomics, transcriptomes, proteomics where the protein profiling, and metabolomics where you study about the metabolites, because metabolites play a major role, because some metabolites are pharmacologically active when compared to the original molecule. And lipidomics, like lipid diversity and pathways, this also play a major role in the drug discovery process. And glycomics, where the glycome study, glycan protein interaction that will be studied. So this all involved in the biotechnology process. So when you study a biotechnology, you can link with all these things. So when you study about genomics, you can directly link with the drug discovery and development process, how this genomics is play a major role in the personalized medicine. Similarly, the pharmaceutical scientists are prepared to take advantages of omics technology. These are the omics technologies where you can use it in the drug discovery and development. And also now we are also focusing on the sequencing of human genome. Is one of the largest scientific achievements has been done, and that opened the domain of medicine and biology. Now there are many researches going on human genome related medicines. And the secret of human genome and other technologies which can be used to edit the human genome, which brings as a major shift in medical research and clinical practice as well. So this also aims towards an understanding of disease. So we can understand about what is the disease, the targeted disease, and based on that, what is the etiology is involved in that. And accordingly, you can come up with a personalized medicine at the molecular level. Because this is the now trend actually, the personalized medicine, which compared to the medicine which can be used for the treatment of everyone, that is normal drug discovery process. Personalized medicine is mainly based on the genomic based, human genome based, gene based studies. So next will be, I'm going to discuss about the pharmaceutical chemistry. What is the role of pharmaceutical chemistry play a major role in the drug discovery process. As you know, pharmaceutical chemistry means you study normally from organic chemistry, where you study about the functional group, namings, then followed by when you move into the next semester, you may study about advanced organic chemistry, where you study about the synthesis procedure and also the mechanism which are involved in the synthesis. Then later, when you move into the medicinal chemistry, where you study the structure, how it is related to the pharmacological activities. So here I given one simple example. See, for example, morphine, which is having the hydroxyl group here, when it is converted into a codeine, which is methoxy group. So the difference is hydroxyl group is converted into a methoxy group. So this is morphine and codeine. But when you look at the analgesic action, the morphine is 10 times higher when compared to the codeine. But when you look at the chemical structure, there are small, slight modification only. Only the hydroxyl group is converted into a methoxy group. So that means the chemical structure play a major role actually. If you modify the one functional group also, that may play a major difference in the pharmacological action. So that is what you study in the medicinal chemistry. What is the relationship in the structure and pharmacological activity, where you study about structure activity relationship, and also means which including qualitative and quantitative structure activity relationship. So not only the structure, which also the isomers. So as you know, isomers means same molecular formula, same structural formula maybe, but maybe different in the conformational structure or geometrical isomerism. Okay, so here I given one more example, levodopa, which can be used for the treatment of Parkinson's disease, and dextrodopa. So here, when you look at the chemical structure, the levodopa is levorotatory. If you look at the chemical structure, it is same. Molecular formula is same. Molecular weight also same. But when you look at the pharmacological activity, levodopa can be used potentially for the treatment of Parkinson's disease. But when you look at the dextrodopa, it is completely inactive. Means what it indicates? Because the chemical structure is same, molecular weight is same, and also molecular formula is same, but only the conformational isomers that is different. But that also play a major role in the pharmacological action. So that is what you study in medicinal chemistry, and medicinal chemistry play a major role also in this aspect of drug discovery and development process. And apart from this, where you can get the normally the molecule directly, you cannot simply cannot make one new molecule. So either it may be obtained from the pharmacognosy from the natural source, or in the previous discussion I mentioned about the pharmaceutical biotechnology, you can also get some of the secondary or primary metabolites from the microorganisms. So that will be, once you identify the chemical structure, then only the pharmaceutical chemistry person will identify or determine with the synthetic procedure how we can synthesize that particular molecule. So once they done the synthesis, then they can, before moving even into the synthesis part, now they have the software like computational chemistry, where they can identify the structure activity relationship before when they move into the in vitro and in vivo studies. So you can predict whether that particular molecule is, how much it is interacted with the enzymes, or maybe the proteins, or maybe the receptors. So that can be determined nowadays in the by using the softwares, which is in silico studies, where you can do the docking studies. You can determine the pharmacological activities as well as the biological activities, some of the biological activities, by using the software. Because previously, even I think 10 years or 15 years before, when the people are started up doing any kind of synthetic related work, what they do normally, they synthesize 50 compounds or 100 compounds. Then what they do, all the 100 compounds they take into a in vitro studies, and from the in vitro studies they select a few compounds and they move into the in vivo studies. So it is something like a time consuming process, as well as very expensive, because we are testing for the large number of compounds. So how we can minimize the compounds to test for in vitro and in vivo studies? By using in silico studies or molecular docking studies. So if you do that one, then you can reduce the number of compounds that is need to be tested for the in vitro and in vivo studies. Because nowadays animal ethical committee approval also is based on the results that you may get it from the in silico studies, because simply you cannot test some new compounds to the animals, because animal ethical committee also very strict nowadays. We cannot simply use or sacrifice the animals without knowing whether that particular compound, whether it gives some kind of pharmacological action or not. So that can be determined by the in silico studies. So here only the pharmaceutical chemistry person or medicinal chemist will play a major role, and these are all the things that you study in a Bachelor of Pharmacy course as well. So as I mentioned, the pharmaceutical chemistry also essential, especially to understand about the structure activity relation studies at the initial stage of drug discovery process. So where you can also determine how the interaction with the enzymes, proteins, okay, or maybe you can select the molecule. It is not necessary that you need to take all the molecule into the in vitro or in vivo studies. Selective molecules, and based on the even you can determine the toxicity profile also. Okay. Like molecular modeling and molecular imaging techniques. There are so many new techniques are available nowadays in the bioinformatics side. So that can be utilized by the pharmaceutical chemist. And for the faculty members, what I would like to mention in this point especially, so in our syllabus, there are latest so many technologies are available. So what in the current syllabus we need to include? We need to include all this latest technology, so that when the students study about all this technology, once they completed their B Pharm, they will get some idea where they can go for their career. If they choose R&D, or maybe if they choose postgraduate study, they can choose which specialization that they are interested and how that can be related with the drug discovery and development process. So hence, with prediction through bioinformatics studies, as I mentioned, we can design a specific ligand or the lead molecule that could reduce the research cost, time, and resources, because we can reduce a lot of time if you do the in silico studies. And also nowadays, one more recent technique is radioligands. So radioligands is something new for most of them, which can help in determining the receptor density. Normally in the in silico studies or molecular docking studies, you can determine how much the efficacy that particular compound or maybe the molecule which can having the binding capacity with the proteins or maybe the enzymes. So here we can determine the receptor density and receptor occupancy, also pharmacokinetic properties, if you use the radioligands, which is a new method which you can adopt to in the drug discovery and development process. So apart from that, in pharmaceutical analysis, as you know, mainly pharmaceutical analysis play a major role in the qualitative and quantitative analysis. So everywhere the pharmaceutical analysis play a major role. If you are can be done by using chromatographic techniques, and you can purify by using some of the chromatographic techniques, which including column chromatography, HPLC, gas chromatography. These are all the chromatographic techniques you can use it to isolate and purify the compounds. So once you isolate and purify the compound by using some of the chromatographic techniques, it moves into the characterization part. So where the infrared spectroscopy is mainly used to determine the functional group, NMR is mainly used to determine the protons and carbons. Then mass spectroscopy, where you can determine the molecular formula and molecular weight. And so finally, you can come up with the final chemical structure of the isolated compound, or maybe the compound which are isolated from the microorganism. So structural characterization is very, very important. So that can be done by using all the instruments. So where you study about all this instrument in the pharmaceutical analysis subject. Okay, so that is what all these instruments like HPLC, GC, UV, these all play a major role in the quantitative analysis. If you want to determine how much quantity is present in that particular fraction, that can be determined by all these techniques. Similarly, some of the pharmacologic and toxicologic related studies can be done by using LCMS and LCMS-MS, like bioequivalent studies. So that is what pharmaceutical analysis also play a major role in the drug discovery and development process. So as I mentioned, 3 pillars in pharmaceutical analysis, which including identification, which is qualitative analysis, identification and characterization, then followed by quantification, which comes under quantitative analysis. So which starts from the starting material until the final drug candidate. So for example, if you start isolating some compound, you need some help from the pharmaceutical analyst, who can help us to operate the instrument and get the spectrum need to be done, and later on you can do the characterization. So from the beginning stage until the final drug discovery, the drug candidate stage, which is bioequivalent studies, we need those kind of instrument. So which is clearly indicating that the pharmaceutical analysis also play a major role in the drug discovery and development process. And here I mentioned some of the latest technologies which you can include in the syllabus, or maybe, I don't know, maybe in some of the university syllabus, I think they already included about these kind of new technologies or new techniques which has been used in the pharmaceutical analysis. So the advancement of technologies and their implementation with artificial intelligence in machine learning techniques. So nowadays we have sophisticated instrument, but still when I look at some universities, they're still using the old one, very old instruments. So if you use those kind of instruments, the problem is you are far behind to the research actually. Because if you look at the Western country people, if you look at their even research publication, they have been adopted and they have been used all the latest technologies, instruments. But we are far behind in research. Why? Because we are not using the latest technologies. We need to provide all the sophisticated instruments to the students, so that definitely they learn, and when they go for a job, then easily they can adapt with the instruments which are available in the industries. Okay. And apart from that, there is a new software that is, I think most of them might be know about this one, analytical QBD, which is quality by design. So where you can design and determine all the analytical methods, development and everything. Because previously we used to do everything manually. Now we have the software which can show a pivotal role in the current analytical techniques in the pharmaceutical approach. So nowadays we are not doing all the manual, the method development and validation and everything. Now everything we have a software, and especially this software, the analytical quality by design (QBD) play a major role in the method development and validation process. So that is what I mentioned in this picture. This can be used for the method validation, method transfer, chemistry system screening. So this has been used for various things. So these kind of things only we need to be included in the syllabus of pharmacy course, so that the students will be know about this kind of softwares. And when it moves into the pharmacology side, as you know, pharmacology play a major role actually in the preclinical side, which starts from the in vitro pharmacology, which including the either you may use chemical methods, or maybe you may use some cell lines, or maybe the tissues which can be used to determine the biological activities, where you can determine the IC50, the concentration which shows 50% inhibition, that is comes under in vitro pharmacology. So after that, in vivo pharmacology, which is using the animal models, where you can determine the drug metabolism and pharmacokinetics, DMPK studies, and also where you can study about the bioavailability, and the safety pharmacology, which including both, there are so many in vitro and in vivo methods are available to determine the safety of the molecule which you are testing for the pharmacological studies. And apart from that, if you move into the clinical side, we have the PBPK, physiology-based pharmacokinetic studies, which are recently used, and the quantitative system pharmacology and qualitative, the quantitative system which can fix appropriate dosage form. You can determine the dose, how much dose can be used when we move into the human studies. And these are the skills that you as a student, you must know as a pharmacist. So if you study all this, and at the same time you must learn all these skills, so this will leads into your role in a drug discovery and development process, which including as I mentioned, the in silico methods, all the computational tools, and all the softwares which are currently available, and also some of the design of experiments, animal handling, statistical analysis. So these are all the skills are required to play a major role, especially in the drug discovery development process in the pharmacology side. So pharmacologists better understand pharmacokinetic principles, which they can be considered more suitable for conducting drug metabolism and pharmacokinetic studies. And once the new chemical entities passes the bioavailability test, where we determine how much amount of drug which enter into the systemic circulation. So the most important part of preclinical research is inducing the disease in various animal models that can mimic the human disease. So your main important thing when you select the models, don't select some models which are not mimic to the human disease, because our main aim is we need to come up with a drug molecule to treat for human mainly. We are still doing for some research on veterinary related research, but at the same time, our main focus is on to come up with a new drug molecule for the human disease. So when you choose the animal models, you need to choose the animal model that can mimic the human disease. So that way, whatever the study that you do, it is appropriate for the drug discovery and development process. And another thing is the safety pharmacology studies, which including the where you can use it cell lines and tissues. So this is what you need to learn actually during the your Bachelor of Pharmacy, or those are doing postgraduate studies. Instead of directly going for the in vivo study by using the animal models, you can do first with the in vitro studies. And also there are some in silico studies also you can conduct, where you can determine the toxicity profile of any molecule. So on the other hand, PBPK, which is physiology-based pharmacokinetic studies, which can helps to make research and development decisions relating to the clinical trial design. Even we can determine first in human dosing. So you can determine the dose, how much dose can be used for the human. And you can also, based on the results, you can also determine which formulation may be suitable. That also can be determined. And dosing in special populations, that also can be determined, because when you study with all these models, you can determine which dose may be suitable for which type of populations. That also can be determined. And prediction of drug interactions or drug food interactions, all the these kind of information also that you can get it from the pharmacological study. So when you study pharmacology, you definitely you study everything, but you are not directly linked with the drug discovery and development process. You need to link what are the subject you study, how that is related to the drug discovery and development process. Because even if you ask some students, especially the first year students, so when I ask them what is the use of studying organic chemistry or physical chemistry, sometimes they may not be able to tell exactly how it is related to the drug discovery and development process, because each and every subject or every course that you study in B.Pharm, which is directly related to the drug discovery and development process. Because, for example, organic chemistry, you study only the chemistry, what is the name of the compound, what are the functional group. But as I mentioned, when you study later on in medicinal chemistry, you can link how that functional group is play a major role in the pharmacological activity. So every functional group play a major role. So as a pharmacist, you must know what all the functional groups are present, how electron withdrawing group, electron donating groups, hydrogen bonding, ionization, everything you must know. You study everything, but you doesn't know exactly what is the use of those things when you study. So don't simply study any subject. You directly link how that is related to the drug discovery and development process, so that you will find some kind of interest also when you study that particular subject. So next, the final one is about the pharmaceutics, where the pharmaceutics subjects play a major role in the drug discovery and development process. So here, when you look at pharmaceutics discipline, you study normally many subjects, which including you study physical pharmacy, where you study about all the physical properties of drug molecule, particle size, size determination, diffusion, dissolution. So this all you study in physical pharmacy. So this all play a major role in the drug discovery and development process. Okay. And similarly, you study about pharmaceutical engineering, where you study about all the instrument and instrumental to make blending, granulation of the material to make into a final dosage form. So because you need to make certain molecules to be ready to make it into a certain dosage form, so you need kind of instruments. So where you study in pharmaceutical engineering. Similarly, pharmaceutical product development can be development of dosage form, characterization of dosage form, optimization. This all you study in the pharmaceutical product development. And another thing, biopharmaceutics and pharmacokinetics, where you study about what are the factors which can affect bioavailability, the mechanism and kinetics of absorption, distribution, metabolism, and excretion. And you can also study about the determination of the dose and dose frequency. So these are all the things that you study in the B.Pharm, which are related to the each and every step of the drug discovery and development process. Because pharmaceutics normally play a major role in the final stage, but their role is very important, because they are mainly involved in the final dosage form selection. So the pharmaceutics normally deals with the presentation of the drug into a patient-friendly dosage form. So there are many dosage forms are available, including tablet, capsule, suspension, cream, lotion, ointment. So many formulations are available. Okay, so that all where you study in pharmaceutics only. So physical pharmacy, pharmaceutical engineering, pharmaceutical product development, biopharmaceutics are different branches of pharmaceutics which also play a major role in the drug discovery and development process. So the feasible dosage form, the route of administration and time of administration are the contents decided in this specialization. So when you study all this subject, you can learn about what is the dosage form which may be suitable for that particular molecule. And also you can also giving the information that which route of administration will be better based on the drug delivery systems. So the pharmaceutical dosage form, which including tablets, capsule, solution, suspension, emulsion, semi-solid dosage forms, which including cream, ointment, sterile dosage forms, controlled drug delivery system, and specialized formulations such as nano formulations. So nowadays, nanotechnology also play a major role here. So we can convert your molecule into a nanoparticles. So that can be used for the preparation of many different dosage form, and which will be very useful, particularly to the drug delivery systems. So apart from these 6 disciplines which I have been discussed in my presentation, there are certain latest techniques also the researchers normally they follow to develop a new drug molecule, especially drug repurposing. So this also some of the researchers, I think degree students also might be knowing about this one. So this is a very latest, I think maybe in the recent 5 years, there are many researchers they adapted with the drug repurposing. So to minimize the time of drug discovery and development process, because as I mentioned, drug discovery and development process takes minimum of 10 to 15 years of time to bring one new molecule into a market as a drug form. So if you adapt with the drug repurposing, within 1 or 2 years, you can come up with that drug molecule for the treatment of certain diseases, because you are using the already existing drugs and you are using for the different purpose. Means previously maybe the drug might be in the market already available. It might be used for the treatment of diabetes. Now maybe we are coming up with the you study that particular drug for example cardiovascular related disorders, and you find that is very effective. Then you are coming up with the solution for the heart related diseases. So this is called repurposing. But repurposing, there are several stages are involved. You need to learn, you must get all the literature about that particular drug molecule, and you need to do a kind of evaluation, then structural analysis of the drugs. Then you need to conduct some of the preclinical studies. But approval is very, very easy for the repurposed drug, because that is already available in the market, which is already approved by FDA. So those kind of drugs very easily we can bring into a new form for the treatment of other disorders. Okay, so drug rescuing or repurposing are offering pharmaceutical companies and the academic world useful and profitable opportunities to discover novel and innovative therapeutic applications. So this way you can come up with so many new molecule, or not new molecule, it is an existing drug molecule, but which can be used for the treatment of different disorders. Okay. And apart from that, another one more thing which you can adapt is translational research. So translational research which comes under innovations. So translation has become a buzzword in the field of biomedical sciences, including pharmacy. So previously, when we do research in the pharmacy discipline, we don't focus much on with other discipline like engineering side. We don't focus. We don't come up with something. We don't think about innovation and all. But nowadays, many researchers they started off thinking about innovation. So innovation, there is no boundary. It is not necessary that you need to conduct several clinical studies, and it won't take much time. Also, like 10 to 15 years of time to innovate something. So this way, you can identify a significant gap in R&D practice, because R&D is normal research and development takes a lot of time to conduct all the studies and everything. So those are thinking that is not feasible. Then you can adopt with the innovation model. So where you can think about something out of box. Okay. So pharmacy means it is not only you need to come up with only the drug molecule which can be used for the treatment of disorder. You can come up with a new kind of device or instrument which can be used for the patient purpose, because we are more towards to patients. So whatever they need, especially that need to be fulfilled as a pharmacist, means we can also design and develop a new devices which can be useful for the patients especially. So implementation of new ideas into practice, that also very important. Even here in Malaysia, in my university, so we have been developed many innovative products which can be used for the especially for the treatment of certain disorders. The simple formulations that we have been made up with natural products, and that is already even some is even still currently available even in the market in the different dosage form. So you can adapt with the innovation techniques. Okay, if you feel something drug discovery and development process is a lengthy process to come up with something, so make sure the knowledge or technology which is produced is useful and relevant to the intended customer. So our main aim is we need to satisfy our customer. Who are our customers? Maybe their patients or maybe the general public. So if they want to use our product, we need to satisfy their whatever they need exactly. So we need to identify what they need in the market, what type of alteration that they are required. Okay, so if you know that one, definitely you can come up with so many innovative products. So scientific approaches that are carefully crafted to connect a lab and real life circumstances as closely as possible. So that is why nowadays we adopted with so many innovation techniques. Okay, which is patient friendly and also the customer friendly one. So I think I already running out of time, so I move into my final conclusion and recommendation to the future pharmacists, those are studying B Pharmacy, and the current pharmacists, our faculty members. So apart from the professional integrity of pharmacy, we are studying more towards a multidisciplinary subject. We are not studying only the chemistry, we are not studying only the biology. We study everything, pharmacology, pharmaceutics, means from the beginning stage until the end stage of the drug which reaches to the customer from through retail pharmacy. Pharmacists only play a major role. Identification of new molecule, pharmacists play a major role. What are the preclinical and clinical studies has been done mainly by the pharmacist. So pharmacist deals with all these areas of drug development, starts from the preclinical to clinical research and patient compliance. So pharmaceutical researchers have been involved throughout the drug discovery process, which including transition of drug candidate from preclinical research to clinical stage. So that is why I told we need to be proud actually for being a pharmacist, because and also the scope for pharmacists is very wide actually. Okay, so you can work in industry, you can work in academic, you can in industry, in R&D. There are plenty of opportunities are there, but the thing is you must be adapted, or you must know about all the latest technologies. So that is what I would like to inform to the faculty members. You need to include all this when you teach to the students, so that the students, they know what are the new techniques are available in the drug discovery and development process, so that once they learn, they automatically get an interest in that one. And when later on, when they move into postgraduate study, or when they enter into a industry, they can do all this kind of work actually, and they can come up with something new, innovations or everything which can shorten the drug discovery and development process. So the global role of pharmacy in drug discovery and development, and shed light on each subject role in future drug discovery. So that is what I have been mentioned, how all the specialization are related, and how each and every subject that you study in Bachelor of Pharmacy is how it is related to the drug discovery and development process. So as a pharmacist, when you study all the subject, you must have direct link with the drug discovery process, because what are the subject I have been mentioned here, everything is related to the, especially for the preclinical side. So apart from that, that's all from my side. So I feel myself proud to be a pharmacist. I hope my presentation might be very useful, especially for the young researchers and also the future pharmacists who are studying Bachelor of Pharmacy currently. So That's all. Thank you very much to everyone for your patience and listening my presentation. I think the time is very limited and it is very difficult to explain each and everything. And my purpose is I want to include all the subjects which are included in your Bachelor of Pharmacy, so that you come to know how the pharmacy education, whatever you study, what are the subject you study, you must know what is the use, what is the function of that particular subject, why you study organic chemistry, why you study about pharmaceutics, why you study microbiology. So everything is related to the drug discovery and development process. But later you can choose your career which you are more interested. But as a pharmacist, when you study a Bachelor of Pharmacy, you are studying multidisciplinary subject. So multidisciplinary subject which including all this, uh, we are the best actually compared to any other professional course which is available all over the world, because we are studying a lot, especially for the mainly for the uh, drug discovery and development process. Okay, so with that I conclude my presentation. I will hand over to the organizer for any Q&A, question answer, if anything. Then if any questions, I am happy to answer.
Okay, sir. Thank you, sir. Thank you very much for your valuable speech. Participants, if any question, please type in the chat box. Dear sir, okay,
Can I just, can you give me 5 minutes?
Okay, sir. Okay, do it.
Okay. Thank you very much. Let me introduce myself. I am Professor Dr. A.K. Gyanachandran. Am I audible to all the audience?
Yes, sir. Okay.
Yes, sir.
Okay. It was a very excellent presentation by my pharmacy colleague, Dr. Mahendran Sekar from Malaysia. He has given a very excellent inspiration speech about our pharmacist functions in all over the world in global perspective. He has covered all the areas of our studies: pharmacology, pharmaceutical biotechnology, pharmaceutical engineering, pharmacology, many things. Everything is excellent. Let's add one or two additional points. So nowadays, this COVID-19 is going on, pandemic, and the DRDO has come to drug. But later they found it is having a more blood supply to the genital organs of the male. So they started using for erectile dysfunction treatment. Similarly, all of us know about the thalidomide episode. Which you studied in pharmacology, and basically it has got lot of side effects like teratogenic effects. Now it is repurposed for treatment of epilepsy. So like that, many molecules are there which can be repurposed. These are the awareness for our young pharmacy students, and as well as our pharmacy young faculty members. And I like that one more thing, because of this latest vaccination going on, people attitude has got some lot of allergy manifestations, and there may be some chances of anaphylactic shock. Anaphylaxis, normally they go for a worldwide EpiPen, that is epinephrine. Now we are as a pharmacist, we are going to formulate normally when EpiPen self-injection. Am I right, Mahendran Sekar?
Yes, sir, you are right.
EpiPen is normally a self-administered anti-anaphylactic agent, but many people, they are afraid of taking this. So we are now going to come out with a sublingual dosage form of adrenaline.
Yeah.
We are doing research in Bangalore, either in a sublingual dosage form of adrenaline or a buccal film of adrenaline. So like that, we have a lot of opportunities. As Dr. Mahendran Sekar has informed, this our discipline, pharmacy, is a multifaceted and multi-oriented approach. The main center is for the benefit of patient. We are cooperating with the patient. These are the few additional points I am adding to the welfare of my students, as well as our new teaching faculty. So thank you very much, Dr. Dhanapal, for giving me the presentation of 5 minutes of my opinion. And thank you, Dr. Mahendran Sekar. Welcome. Thank you very much.
Thank you, sir. Thank you,
for
sir.
your opinion.
Thank you for your valuable information, sir. Thank you, sir. Finally, we goes to vote of thanks by Mr. Gnana Sekaran, Associate Professor of Sree Sastha Pharmacy College.
Thank you, sir. Thank you, sir.
Thank you to it.
Hello, everyone. Very good afternoon to one and all. Myself, Dean Gnana Sekaran, Associate Professor, Department of Pharmaceutical Chemistry and Analysis, Sree Sastha Pharmacy College. It gives me an immense pleasure to deliver the vote of thanks for this webinar with this inspirational speech and as well as a valuable time. And I would like to thank our beloved chairman, Professor J. Karthekeyan from our Sree Sastha group of institute. And my special thanks to Dr. John C. Charles, former Register of Tamilnadu, Dr. MGR Medical University, Chennai, and active participation from this webinar series. Thanks to Dr. Samuel, Principal of Sanju College of Pharmacy for his valuable participation. Once again, my special thanks to all the participants, including principal, professors, and as well as associate professors, assistant professors, and students, as well as our Sree Sastha Pharmacy College staffs and students, and the participants from Tamil Nadu. So, of course, not only from Tamil Nadu, from other states participants, especially from Uttar Pradesh and Gujarat. So, in and around the world, pharmacy professions and as well as other healthcare professions. And my final thanks to all the members and as well as participants for this webinar is for a great success. Finally, I conclude with this proverb which says, "Noetra valve, kurevatra selvam." So, health is wealth. So, have a grateful and as well as a great day. Thank you.
Thank you, sir. Thank you, sir. Thank you, one and all. You collect your certificate, and I already given the link. So you go to this link, you collect your certificate. Thank you, all the participants who actively participate this very much.
Thank you.